Hello and welcome to my video on ethyl alcohol fermentation. Today we'll be talking about the metabolic process known as ethanol fermentation and how, why, and where it occurs. What is ethanol fermentation? Ethanol fermentation is the creation of energy from glucose, with the end product being ethanol, which is the alcoholic content seen in beverages such as beer, wine, vodka, rum, etc. Why does it occur? It occurs in order for organisms living in an anaerobic or oxygen-free environment to produce energy in the form of ATP. Inorganic alternatives. As far as I can tell, none. It would be insanely inefficient for alcohol companies to research and develop a way of making ethanol inorganically when they have an army of microorganisms who will do the work for free. In which organisms does it occur? The process of alcohol making, and by extension ethanol fermentation, utilizes tiny fungi known as yeast. There are many strains of yeast, but more on that later. Dangers and benefits. There are little to no dangers in the metabolic process itself, due to the fact that it naturally occurs in nature and is the result and has been perfected over millions of years of evolution. There are some dangers, however, in humans utilizing this process to make alcoholic drinks. If the container that has yeast in it does not have way for carbon dioxide to escape, the container will become pressurized and eventually explode, causing lots of property damage and potentially harming workers depending on the size and material of the container. The benefit is alcohol is a hugely popular, very profitable product sold almost worldwide. Who doesn't love alcohol? Final question. How does alcohol fermentation occur? Is the main focus of this video and will have to be explained in great detail. Fermentation first begins when the yeast are introduced to a container filled with oxygen and a substance which will provide them with glucose. For example, if you're going to make wine, you'd add grapes as your glucose-giving substance, and if you're going to make mead, you would use honey. The container containing some oxygen may seem odd at first because alcohol fermentation only begins in an oxygen-free environment. Believe it or not, the oxygen is actually beneficial to the brewer, as the yeast can undergo normal cellular respiration and thrive, reproducing rapidly, until the oxygen is depleted. Once the oxygen is depleted, there is a large colony of new yeast which can undergo alcohol fermentation at a greater rate than the yeast could initially. After the oxygen is depleted, the electron transfer chain no longer has its final oxygen electron receptor, and the system backs up. Therefore, the only way the cell can produce energy is by glycolysis, which is okay until the limited amount of NAD+ is completely converted to NADH. You can imagine it as sort of a roadblock or wall being built after pyruvate oxidation and before pyruvate oxidation, after glycolysis, by oxygen or the lack thereof. So without oxygen, ain't nobody getting past this wall. The pyruvate can no longer continue down the path of cellular respiration and have to take a detour down ethanol fermentation road. Welcome to ethanol fermentation road. This is where the actual process of ethanol fermentation happens. So to start off with, we take our two pyruvate molecules, which were formed during glycolysis, and we turn them into two acetaldehyde molecules. And this is done by removing the terminal carboxyl group, giving it off as CO2, and we're left with our acetaldehyde molecule. So we go from 3 oxygens to just 1, and 3 carbons to 2. And the enzyme that facilitates this process is pyruvate decarboxylase. Notice decarboxyl, as in removing a carboxyl group. So after we've got our two acetaldehyde molecules, we reduce them into 2 ethanol molecules using the enzyme alcohol dehydrogenase. So we start off with this, and we add an hydrogen ion, and we end up with this. We know from our experience with redox reactions that if something's reduced, something else must be oxidized at the same time. In this case, the 2 NADH molecules formed by glycolysis are oxidized to 2 NAD+ molecules, which can then travel back and undergo glycolysis again, producing more energy for the cell. Because as we remember, the Krebs cycle, electron transport chain, and the pyruvate oxidation has all been blocked up by the lack of oxygen. So this is the only way, glycolysis is the only way that the cell can produce energy. And what's interesting to note about this entire process is that ethanol cannot be used by the yeast cells at all. It's in fact toxic to it. So the only reason it produces ethanol is for this redox reaction right here, which oxidizes NADH to NAD+. So ethanol being toxic is also the reason why most alcoholic beverages have a 15 to 16% alcohol rating because the strain of yeast used by most brewers can only withstand 15 to 16% ethanol before they start dying off. But there are more strains of yeast that can survive in higher percentages of ethanol, which, for example, a yeast that is more tolerant to ethanol can be used in vodka production, while a yeast strain that is less tolerant to ethanol will be used in a beer or something. So for a final sort of overview, yeast, which looks like that by the way, tiny little grains, and these are all fungi by the way. So these tiny little grains plus glucose, which can come in any form from grapes, you can have wheat, you can have honey in some cases. Any of these, as long as they contain some glucose for the yeast to turn into energy, will produce beer, wine, any alcoholic drink, anything that has the ethyl alcohol that looks like that, aka ethanol. As you can see, there's an alcohol group at the end. So yeah, that's all. Thanks for watching.